Good afternoon, all of you. We'll be having our next session by the speaker who has speak yesterday. He's Dr. Shiva Kumar. So I already introduced him, and he has done a very splendid lecture yesterday on the AI on drug discovery. So today he'll be talking about the another aspects, that is the targets, therapies, and mostly the side effect challenges. So by experience, he is a medicinal chemist, also an AI expert. So he's the founder from that Biocognize, and presently they are a company that is like the working on the AI for the pharmaceutical development and all. But in profession and the experience, he's actually medicinal chemist. So he'll talk about the topics today on this particular thing. So I think Dr. Shiva, you can start the presentation. Just speak something. You have to make it on. Make that.
Everyone, it's my pleasure to meet you guys again on the second day of the conference. So today we are going to focus on the side effect, mainly in a drug discovery journey. In yesterday's sessions and today's sessions, from various lectures and from me also, you might got some idea on how the drug discovery journey works, right? And what is the drug discovery pipelines comprises of in both experimental side as well as on the computational side. We focused more on the AI part yesterday, along with the parallelly, what are all the alternatives for CAD, computer-aided drug discovery, as well as for the AI-based drug discovery. We have seen various tools and techniques used in both computer-aided drug discovery method as well as AI drug discovery method. So today's lecture, I will quickly skip, I will talk about those things again, but I won't dive deep into that. Instead, I will focus more on the side effects and how we can proceed further. Okay, so this is the drug discovery pipeline which we also seen yesterday. Yeah, so if you look more deep into that into the drug discovery journey, so the main key steps involved in this is identifying the target, then identify the compound, and then the testing of the compound. Right? This is very broadly you can classify in drug discovery pipeline. So the main important thing is on identifying the target. The target here, I mean, is usually biological target, right? It's going to be any organisms For example, if in the case of COVID, we have the external virus in protein, right, like that. Or sometimes it's going to be a host itself, human itself we will do. For example, disorders like diabetes, cancers, where we will target the human itself. Like when there is a host pathogen interaction, sometimes we will work on the interface also. Some vector-borne diseases like that. Okay, so it varies, but usually the targets means the biological target. We generally mean usually protein, but not always. Sometimes we'll target DNA and other macromolecules also, right? So there are a lot of methods to identify that one. Using computationally, we use genomics and proteomics approaches. Bioinformatics plays a huge role, and at Biocogniz, we are majorly focusing on target identification and lead identification using next-gen sequencing, data analysis, a team of AI experts and data science scientists. We majorly work on identifying the targets from the big data. For several diseases and disorders, even though we are majorly focusing on cancer, but we are not limited to cancer. We are working on multi-targets. And then we focus on the compound screening, right? Once you identify the target, the next important thing is how you target it. The target, what you will target with, usually the small molecule, but not always. Sometimes we go with the macromolecule also. Sometimes will be a peptide, sometimes peptidomimetics. But in general, the compound, or if you say a drug, it usually means a small molecule. It's chemical substances, but not always. Okay, where it's why we are talking about side effect here is because of we are individuals, right? Like in the case of target identification itself, when you identify a target, it's about whom you are targeting. But in general, when you are working on when you are working on the multi organisms or multiple individuals, so it will vary. Actually, it won't be the same, right? Because genotypically and phenotypically we are different. So that's where the pharmacogenomics play an important role. Individual, right? So where the NGS also plays an important role. So the side effects comes there, as well as we have a lot of similar proteins. When you target one particular protein, lot of protein, we have a homologous protein and also the similar proteins, structurally similar proteins. So when you target one protein, the other protein will also be get targeted. There where the side effects comes, and the selectivity plays a very important role. And then once if you can able to achieve it, then we will move on to the preclinical and clinical trials, and finally it comes to the market for the clinical use. So this is what the drug discovery journey comprises of. Okay, so we're all talking about drugs, so we should know also what the drug is, drug mean. There are a lot of definitions. If you just Google, you will get a lot of definition for a drug. But according to US FDA, the definition of drug says that it is a substance intended to use in the diagnosis and cure. This is where majorly we are focusing on, and also to mitigate the effect, so reduce down, or also the treatment, and sometimes for the prognosis also, as a preventive method. Also we will use this like prophylaxis, right? Like vaccines kind of thing, you can say. Vitamin B complex is one of the best example, and sometimes for the diagnosis also. Usually for the treatment, majorly, if you think of drug, it comes to your mind is the treatment. Yeah. So now you have a drug, and now the question is like yesterday in the previous lecture we have discussed about how and all we can design and find the drug, right? So if you can able to make a drug, the next important question is, okay, you are targeting the target of interest, fine, but why we are getting side effect? You know what, like as of now, if you just go to pharmacy and if you can get any medicines, and you can just check the pamphlet, you can see that it will all have some kind of side effects. There is no drug as of now available which has zero side effect or no side effects. To me, the difference is like less or more side effects, only those things. The problem is because of it's not specific and selective. That is the reason. Okay, so we should know that what is the selectivity and specificity. Selectivity means like it's the ability of a drug it's need to know that how it can discriminate between the related target, right? One it shows to higher binding compared to the another one. Whereas the specificity is it should bind with only with one and not with the other, right? That is the specificity case. So how we are going to achieve this selectivity and specificity? If you can able to achieve this, automatically you can avoid the side effects. Very simple. But achieving this is not an easy task. There are several methods used in the literature. I'm going to show you some of the methods used in the literature and some of the methods I have used in my research. So these are the two things we are going to focus today. So this is just a overview of what we have discussed last class, like in the computer-aided discovery as AI-based methods, whether you can broadly classify into structure-based and ligand-based. When you know the protein structure, we will go with the protein as centric, or if you don't know the protein, we will go based on the ligand-based methods. So we have several methods. We have discussed all this method last class, including the CAD as well as the AI-based method. So I'm just skipping this one. And one of the method we will get the drug, the target therapy or the drug is by visual screening. So we have several methods. All the methods almost have one common characteristic, which is called the funneling approach. That means we start with n number of compounds. N is usually millions or billions of chemical substances or natural products, anything it may be. Usually the small molecule substances, not always though. And from there we will narrow down to fewer number of compounds in order to synthesize and test it at the preclinical and clinical. This is the ultimate. If you look into 1,000 visual screening articles, you will end up only this simple diagram of funnel. Okay, so there are many methods. Some are using a hybrid methods, using different filters, different approaches. Ultimate aim is to reduce the number of things. If you see in this method, they have used both the structure as well as the small molecules. You can say like it's a hybrid approach using both structure-based as well as the ligand-based approaches, and also various filters. And here you can see it also uses to something like scaffold hunters, by means like you can also modify the scaffold once we identified this thing. And in this one we are using some approach called pharmacophore. Pharmacophore is like a feature. Instead of directly considering the atoms, we are representing the atoms into the features like hydrogen bond donor, acceptors, hydrophobic, ring aromatic, like that. And based on that we generate a model, and then we will do this one. So another alternate approach in order to reduce the numbers from billions to handful number of compounds. So again, this is another approach. We have used one more here. Till previous methods, this uses mostly static approach. In this method, we are also uses a dynamic approach by using molecular dynamics. So molecular dynamics is computationally expensive if you use at this many number of compounds. So we will generally use at the later stage. So we can apply some PK, PD, pharmacokinetics, pharmacodynamics, filters, dockings, all these things, and then narrow down the compounds to like maybe hundreds of compounds or thousands of compounds. After that, you can apply, study the molecules in dynamics or in motion. Then you can better understand the interactions and its stability. So that also is possible. This is also another approach where we have used both ligand-based and structure-based, and then various physics-based scoring functions and machine learning-based approaches combined here, both CAD and AIDD, in order to narrow down the compounds. Okay, so overall, we can use various databases, methodologies, scoring functions, docking algorithms, CAD or AI, whatever. But ultimately, what we are trying to do is like mainly whether it's able to get the minimal number of compounds. Again, the question is still remains the same, like can we able to achieve the selectivity? You see, right, even though I quickly browse through the slides, one thing you might have commonly understand is like we have used several approaches, and several approaches containing millions of compounds, some contains billions of compounds also, and from that we can able to narrow down to like 10-20 compounds. So from million to fewer number of compounds, right, which is synthesizable numbers at the lab level. So but the question is, okay, you narrow down by various parameters and it can be tested, but whether the compound is selective, it is free from side effects or no? The answer is obviously no, right? Why, we will see. So these are some of the various approaches, other approaches like de novo design. This also we have seen. Why we are using de novo design? Because we can go from the IP problem. Like in the industry perspective, if you look into in industry, we have we are not looking for just publication, but also we look for it should be patentable, it should be marketable. So if you make something else which is already existing and which comes under the patent, is difficult. So we look into something novel. So one of the best approaches to do that is de novo design, or in the case of AI-based method, we have seen it is generative method, right? So we can use this one, and also we can do a tailor-based method. This method we have discussed already in the previous lecture, like growing and linking approach we can do in order to get the ligand for the desired protein of our interest. But the question is again the same, whether we can able to achieve the selectivity. The answer still remains the same. That is no. Okay. Yeah. And this is the point which I have mentioned in the previous lecture also, but we didn't see that there are several pharma companies, traditional pharma companies. Many things you might have used their drugs also. For example, GlaxoSmithKline company, Merck and Novo Nordisk, and about at least these companies are very, very common, and we all might have used this one. And some more companies like AstraZeneca, which is in India, right? Like Pfizer and Roche, Sanofi, BMS, Bayer. These companies, most of you know it. So these companies are traditional pharma companies. But if you see here, a part of the traditional pharma companies are actually comes into the AI-based role also. They are taking. And there are some pure AI companies like In Silico Medicines and Atomwise, Malevolent, all these things are also exist. So now traditional companies are also comes into the AI-based or the computation. Already they are using computational. Now they are using the In Silico as well as the AI-based approaches, considering the time and cost-effective approaches. They are also moving to. But still some are sticking to these things. These companies, what they are doing is they are not directly doing, but what they are doing is like they are using the pure AI-based companies or the in silico companies, right? So they will tie up with this kind of doing a JV, and then they will work on. So this picture, this is from my article from 2023, but there may be now, there may be some changes might have happened, but not much changes. And there are some companies are going back also because of the risk involved and some of the early stage failures they are focusing on. But overall, so this is the scenario currently in the market. Okay. Yeah, and several AI-based methods. So this is just to refresh your memory. We have already seen this in the previous lecture when you have different method. So we have the overall is like from a training data set. From we will model the make a model, and from using the model we will either generate the compounds or we predict the property of the compound. These are the two approaches. Broadly, we can say that what we are doing with help of AI in drug discovery. Right, so the encoding and decoding happens this way. We encode into some molecular fingerprints from the huge database of our interest which we are using, and then finally it decodes. We'll use some kind of AI algorithms. It's maybe either machine learning based or the deep learning based, and then we can go with it. Yeah, so like this we have several methods are there, but again the questions I like to ask is like whether are we free from side effects, or we can, in other words, can we able to achieve the selectivity? The answer is no, right? So there are several AI-based methods. I'm skipping this because we have discussed this already. Yeah, overall, so now come to the actual question of our today's interest. So that how we are achieving the selectivity. So if you look into the methods which I have briefly described today, and which we clearly, elaborately we discussed yesterday, if you see one thing, that irrespective of the methods and techniques we use, we are not able to achieve that selectivity, right? We can able to get the inhibitor, we can able to find the therapy, we can able to identify the target. It's all fine. But the question is, can we able to identify a compound which is side effect free? Or it is able to bind only with the protein of our interest, not goes and bind to any other protein. If not, why we are not able to find it out? Okay, so that is this thing. So what we are not able to do is, like, maybe you can tell that it is like you need a good chemistry knowledge or chemical intuition, or it's purely a serendipity, or it's maybe it's probability, right? Like it may happen. At what is the ratio? It's that also what it may be. Right. So, but the answer is something very different, which I want to mention it here, is like the answer is not at all on the drug side. It's not on, we cannot blame the small molecule, or we cannot blame the technique. So if you say like, okay, you are using classical docking methods and dynamics, why can't you use some late generation or latest AI-based method? A lot of things are available now. We have DeepSeek now. Why can't we use those kind of highly trained large language models, something like that? No, the answer is not there. Or if you are using whatever the commercial software you are using, or whatever the experimental method you are using, how high resolution you are going on. The problem is at the understanding of the target. Yesterday, I have touched this point slightly, but I didn't focus much on that. That is, we have something called, that is a gap, sequence structure gap, right? The number of sequences available in our hand is huge. And the number of structures we have in our hand, theoretically it's equal. It's like it's almost same, not almost, it is same, because whatever the number of sequences we have in the sequence database, we have structures for all the sequences, but all of them are predicted structures by AlphaFold, like AI methods. But how many high resolution or correct experimental structures is very limited. And they are focusing only on the key important targets only. Not everything is solved. Right? There are constraints are there for that. Why we don't have structure, that is another debatable topic. But whatever the number we are having is very, very limited. It's the huge difference between the number of sequences and the number of structures. So that is a problem. We are not able to. We didn't understand the structures properly. Right? We know the structures, we know the sequences, we know the protein, but we don't know how the structures are. That is also another question. Like, even if you know the protein structure, can you be able to achieve that? That is also another. There are a lot of things. Okay, it's not that as straightforward. But if you can be able to pick up some structures which are more relevant and more closer, then you can able to get into try to solve this problem to the minimal to the maximum effect. Something like this. So the side effects, in simple, how do you define the side effect? That is the question. Side effect is not something which is which comes from somewhere, right? It is for the drug which we are taking. For example, antihistamine, right? If you take cetirizine, or if you take CPM, chlorpheniramine maleate, the older generation antihistamines. The one thing they will say, right? Like when you take those kind of antihistamines or anti-allergic tablets, during the cold time, we will to take this, right? So when you consider that, when you take the tablet, so Avil in commonly known brand names. So if you take that one, what will happen? You will feel drowsy. They will say that don't, if you take this tablet, don't drive or don't come to the class because you will feel sleepy, right? So because of why we are getting that one, right? It's can able to go across the blood-brain barrier and it's go to the target the CNS, even though it has to target the antihistamines. Then the later generation drugs are focusing on the off-target effect. So this is my target protein. But instead of binding only here, it binds here. Along with it, it binds to the off-target protein. So this protein may be involved in some functions. If you block it, usually it's are all inhibitors, right? So if you block it, it produces one effect. If you block this protein, it produces another effect. If you block another protein, that gives another effect. So those effects are we call it as a side effect. But if you are this protein is also needs to be targeted for some time, some reasons. So if you target that one when it is overexpressed, then it is fine. But when it is not needed, when it is not to be targeted, when you are targeting that protein, we call it as a side effect. Okay, if you understand this one, then everything will be clear. So it is not something we can say dangerous. It may be something untowards or unwanted effect at the time. It may be simple as like drowsiness or maybe anything. Antibiotics causes diarrhea, and antihistamine causes drowsiness or sleep, and a headache, nausea, vomiting. You might have seen if you take any drug, anything, if you turn back the strip of the medicine, you will see a lot of side effects. It comes with some are minimal, some are lethal also. They said that sometimes it leads to death also, or many side effects, right, will be there. So that is the reason why we are not able to target this, because we don't know how other proteins will be and whether this protein, where and all it can go on by. So there is a limitation, right? We cannot go and check all the proteins, and also if you find this protein goes onto protein N, protein X or Y, and if you are then you are modifying this drug in order to avoid the binding, then this effect will goes down, activity will goes down. So we need to keep its activity also high as well as the off-target effect low. So that's where the challenge arises. Okay. So there are a lot of methods, like you can go with something like that, identify the chemical structures and identify the probable target proteins and what are all the substituents which we are uses, because most of the time the formulation also causes side effects, not only the API, active pharmaceutical ingredient, the main drug, for example, paracetamol in Calpol or Crozin. That is not the only thing which it has. It also contains many other adjectives. So those additives may also cause side effects, even though most of the times it's all inert. Sometimes that also causes. So we need to find that one also, and then we will look into the effect of this one. And what are all the, you have to see that where and all it causes. And another probable side effect is also causes due to the individual variations. Side effect caused to person X will not cause to the side effect caused to the person Y, because of if there are 10 people in the same family itself, one it gives a headache and another one, the same drug will not give the headache. Why? Because of the genotypically we all vary, right? So that is also another big concern we need to consider. So there are a lot of factors involved in it. That is the reason why we are not able to do this. So we will see how we can this one. So overall, you might have understand one thing from now, like if you can able to achieve the selectivity, you can minimize the side effect, right? So how we are going to achieve the selectivity? That's what we are going to see now for next couple of minutes. So for this, we took a protein called OTB1 and 2, which is structurally very similar protein, belongs to the family of DB-glutinase. So it's a cysteine protease class. Some of you may know cysteine protease is a typical class of protein which contains the signature catalytic triad as the catalytic active site. It also has another unique signature, contains it exists in the active and inactive form. Okay, I will talk about that later in the next slide. So deubiquitination is a normal cellular process which needs to be happen in a day-to-day our activity like apoptosis and other cell differentiation, cell signal. But in the case of overexpression of these OTBs, for example, 1 and 2, which is observed in several cancers. Interesting thing is OTB1, even though they are very close to homologs, or structurally very similar protein, and it is expressed differently in different cancers. In cancer A, it is OTB1 is expressed more, whereas cancer B, OTB2 is expressed. So when you target OTB1, it should bind to the OTB1, not to the OTB2, even though it is very closely similar. So because as I said, the OTB1 and 2 are also needed for the normal day-to-day cellular process. you should not target it unless otherwise it is overexpressed. So we should know when it is overexpressed. That is very easy. A normal blood test can able to identify through the blood biomarkers. You can easily know what is the protein is overexpressed. If you find out OTB1 is overexpressed, but the problem is we are not able to target only OTB1, since OTB2 are also very, very similar to the OTB1. But if you target OTB2 also, because of non-selectivity issues, then it will cause us a lot of side effects. Very simple example. Okay, so now how we are going to do? So the one way is like you, as I said earlier, when we do the work, we did a lot of literature study, and what we found is like previously, everybody focusing on the known structures available in the literature as well as in the databases, where what we got there is like co-crystallized complexes maybe, and the complexes solved in the structures solved in the higher resolutions. All these things we considered, but what they fail to consider is like whether the structure is in the active form or inactive form. Right. So what sort of conformations it is in, that's most of the things fail to address. That is the reason why they are not able to make the drugs into the selective cases. So when we start this work, so we focused on that one. We, instead of focusing on the better active compound, we focused on the identifying the right target, the structure. Right, OTB1, we have several crystal structures, with and crystallized with the complex information already available. So when we studied, so we look into few categories, like whether it's ubiquitin bound or ubiquitin free. As I already told you, right, this is a de-ubiquitinous protein. So ubiquitin bound state and free state, ubiquitin without binding, it is available, as well as it is also available in the two forms, that is in the charged state, because if you look into the catalytic site, it has the histidine and aspartic acid and cysteine, right, these three residues. So you know, right, aspartic acid and histidine are the charged residues, and so we know that whether it is in the charged state or in the neutral state. So the protonation state of the catalytic residues, we observe that one. So when you look into we have to see that whether the catalytic site residues are in the charged states or in the neutral state, and whether in the ubiquitin bound form or in the free form. So these are the two conditions we need to put for both OTB1 and OTB2. So while we study, we consider the distance and angle and other constraints. And if you see here, I'll just show for time constraint, I'm just showing it only the final the confirmation. You can see in the OTB1, it is in the ubiquitin bound protein conformation, and in the charged state, it is active conformer. Whereas in the case of OTB2, it is ubiquitin free, right? It's ubiquitin free, and it's in the neutral state. Right, neutral state, it is active conformation. So, but generally considering the crystallographic constraints or the programs which are using generally at various pH, what we will do generally, we will do at the target pH like 5 to 9. Usually we prepare the protein or the small molecules, right? When you prepare the protein at a particular pH, neutral pH, for example, 7, or at the range pH like 5 to 9 as to basic pH, then you will end up in neutralization, or the charged states of the things will be available. We will not get the exact conformation it's to be. So instead of the active conformer, you may consider the inactive conformer, or inactive conformer state, you consider the active conformer, like that. So you actually miss the information of the nature of the target protein and when it need to be targeted, because in the biological system it exists in this way. So if you are not able to identify this one, the selectivity, you cannot able to get this way. Okay, so that is a very important lesson. This things is like you may refer our JBC paper, like where we have given a very detailed explanation, like how we did all these things and how do you achieve this thing. And also sometimes the water molecules also plays a very important role. understand the selectivity. So we also use when you do the molecular dynamics, we can understand, we can place the water molecules, and we observe that where and all the water molecules travels, and we studied the path of the water molecules, and we found that the water molecules path is also plays a very important role where and all it can able to travel. So that also tells a very important role how this network of interactions plays a very important role in stabilizing the catalytic triad at that particular state. Okay, so that also very important role. So when you target computationally particularly, or through the experimental study also, we generally what we will do, considering the constraint, we remove the water molecule usually, or we will keep only the water molecule which is crucial for the binding. But sometimes the network water molecule, as you can see here, you can see here this is stable by a single water molecule, whereas here if you see in the case of OTB2, it is stable by 2 water molecules. So usually in the docking studies or in the X-ray crystallography experimental crystal structure also, we generally keep the water molecule which will plays an important role in the binding interactions. But we won't consider the interactions which will stabilize the active site conformer. Even if you consider also, we generally ignore this kind of water bridges, more than 1 water molecules, because the distance between these two are far, which is not enough for single water molecule to hold the interactions or make it stable. Sometimes more than 1 molecules will act as a water bridge in order to make the conformation stable and its state in order to bind the other one. So when you design the molecule, if you consider this water molecule orientation and the position, either replace this water molecule at this stage, and then if you make the molecule to go and bind, here we can able to achieve the higher selectivity to go with only this one. Okay. So yeah, so that's what mainly we have observed that one, like OTB1 and OTB2, how we achieve the selectivity is we should know that when it is the catalytically stable state, active state, that we have to identify, because 2 similar proteins, one achieved in the 2 different conditions, either active site the protonation state and the substrate or the peptide binding or the free state, and even the side chain orientation of the active site is also very crucial, and also the water network, not just one single water molecule or crystallographically significant water molecule alone, but also the network of water also we need to consider, which is usually ignored. Okay, so if you can able to do this kind of analysis at the structural level, then we can able to achieve the higher selectivity for the 2 similar proteins. So when you can able to achieve that kind of selectivity, then you can minimize the side effects to this one, right? So we have to go this way. I hope that's fine. Now we extend this one to the host, right? So the human, now what we have seen is only with the human. For example, if you are targeting with the host protein, host is maybe here human, and the antigen from the virus. So what we will do, for example, when we did this time that the COVID starts the outbreak, so we extended this, we looked for the OTB1 and OTB2 for the COVID protein also, and what we found is there is a peptide like protease PL protein which is present in the SARS-CoV, and which is also very similar to this OTB1 and OTB2. Okay, and so previously the previous strain has the covalent inhibitors were available, but the designing of the covalent inhibitor have its own challenges. So what we did is, like, we did a comparison of the viral protein SARS-CoV-2, the OTB1 and OTB2, again the catalytic site analysis. So what we found is, like, it has a very similar architecture of the host as well as the virus. Both are the similar one. Now, if I target the viral protein, what will happen? if I found an PL-Pro inhibitor, okay, PL-Pro is one of the important protein in the SARS-CoV-2, and there are a lot of PL-Pro inhibitors were available at the time. People have started making it and working for it. So if you target the virus with by targeting the PL-Pro on the same catalytic site, this is where it's going to be targeted. But instead of targeting this one, it will also target the host, the human. Right, host in this here in the case is like human. It will also target human also. So here the side effects comes in this way. In the previous case, the side effects comes within the similar protein within the human. In this case, we are targeting the external or the antigen, the foreign protein, which is the host viral protein. But by targeting that one, we will also the side effects comes by targeting in the untoward side effect by getting it targeted by the targeting the normal day-to-day life significant protein like OTP1 and OTP2. Okay, so this one we need to see. So when you design the inhibitor for this one, we need to make sure that one will not bind with the OTP1 and OTP2, or it should not be effective in this one. So selective for the SARS-CoV-2. So we consider both the covalent and the non-covalent inhibitor. That is another big study. So we have considered all the conditions, and then what we did is like again we have used again the water molecules role and again fluctuations and the orientations of the side chains and how much it is varies. Again, mostly the focus is on the target side, not on the small molecules. Okay, so then finally we achieved that we found that the inhibitors which we can what is the main significant differences between the SARS-CoV-2 inhibitor and if you over the PL-Pro, OTP1 and OTP2, we have identified and also reported by just by understanding the active site. We haven't done anything else on the small molecule side, as you can see in that. Okay, so there may be another case where there are a lot of cases comes with the metalloproteins. Usually in the case of considering the force field constraints, so what we will do is we generally ignore the metal atoms unless otherwise it plays an important role in the binding. So usually the metal ions will not be considered. This is another study. It is somewhat different from the previous one. It's like PDE, 2-3 different cases of magnesium. Like we can see different combination of metal ions systems, and then how each thing plays an important role we have studied. So in this case, what you can do is it's at sometimes when we there are different reasons. Okay, like for example, able to make a complex or make able to study or considering the metal state and many other reasons, generally we will ignore it. Right, we will ignore the metal ions and then we will proceed with just only with the proteins. So even for the metalloproteins also. So we have carried out another study like what is the significance of the metal systems and at particular combinations, and what are all the key crucial residues which plays an important role in binding of the ligand in particular metal systems. And we found that what are all the key crucial residues and the non-bonded interactions for the different cases and metal systems. And then we found that the point what I want to mention here is like when you have a metal ion systems, you also try to include that metal ion system. Usually ignored if unless otherwise it is not directly participating in it. Considering the factor of the force field will not support the binding or studying the interactions. So at the time we are actually missing the effect of the metal ion. So we also need to consider that one. So we also considered different metal ions and different, again, MD level, different interaction energies to see that what is the significance of that particular metal complexes with the ligand, even though it's not directly involved in the binding of the small molecule with the protein PDE in this case. Okay. and we have identified some set of compounds and then we have reported. But the point that is not important here. What we need to understand is like the significance of the metal ions. We should not ignore it, even though it's not directly involved in the binding of the protein with the small molecule. Just stating the reason of non-supportive force fields or other reasons, other constraints with either computational side or on the experimental side. We need to look into the other alternate approaches for that one. Okay. Another interesting study, with this I will conclude this selectivity part. That one is like more into the selectivity issues again, but with the kinase. It's a very interesting category. The kinase is a very broad family of protein where it also comes unique thing called the DFG in and DFG out conformer. In other words, like open and closed conformation. Right, so we need to see that which one is active and inactive conformer. And this is also from my work only. I'm just focusing on this one. So it is like AA based. We have used both AA as well as the computer-aided drug discovery approach. And it's a combination of everything. Like we have also used experimental, QMMM, everything, and right, what we found it like is mainly interesting thing is the DFG in conformer. We have used wild type and mutant. We have used a series of kinases, and then we narrow it down to FLT3. In that FLT3, we have used wild type and mutant. Particular mutant of our interest, D835Y, as you can see here. So while in the wild type, it says that you can see that it is in the DFG out conformer, and in the mutant D835Y, it is in the DFG in conformer. Okay, so that's how how it is in the DFG in and the DFG out conformer. That is another story. But what we will see is like when just because it is a mutant, just because of one residue mutant, it changes the from in active and inactive conformers, or in DFG into DFG out conformer, it is changing. But if you ignore these conformational changes, and if you are just using only the mutations at the residue level or the structural level, if you do the mutations and ignoring the conformational changes, main conformational changes, DFG into DFG out conformer changes, which is usually happening. Like in other cases, we won't consider, right? We will just mutate only the residue of interest, and then we will see. But when you do like that, we actually missing a huge thing, that is the two different, right? DFG in and DFG out conformer, totally different conformation of the protein you will be targeting. Either you will consider only DFG out conformer in the mutant also, or you will consider both DFG in conformer in both the cases. In both the cases, you will be targeting in at least one case, you are targeting the wrong conformation. Am I right? So that is also very, very important. If you do that, obviously we will end up in the wrong binding efficiency, and then it will affect into the selectivity issues will come. So when you consider proteins like kinases, we also need to look into the open and closed conformation, or in and out conformer of the protein, in order to see that one, which is the active and the inactive conformer also. Okay, so that helps us to understand the protein and its nature. Then we studied the binding. After that, it shows a better effect. And also electrostatic interactions can also be studied. Like the electrostatics, the protein is in the charged form, right? Like a lot of charged amino acids we have, and we can also, this is also much ignored part, or not much focused on. So we have seen that in another case where human and mouse TDH protein, where we found that very similar protein again, but the charged surfaces are too different. The binding surfaces, the electrostatic surfaces are actually varies, so by which you can achieve the selectivity. So when you look into that one, we can also look into the amino acid composition, and it contains the charges, electrostatic surfaces, and the binding cavity sizes, and the binding site orientation, and also like water molecules present, So all these things, if you consider, then you can achieve the selectivity. It is not about identify a better inhibitor, identify the high binding affinity compound. So if you are focusing only, usually in the discovery side, if any discovery scientist or anybody who are working on the drug discovery part, what we will generally do is like we will try to identify a better IC50 value compound or ED50 compound or the molecules with the very good docking score or docking energy, right? But we should not focus only on that one. If you are focusing on the selectivity issues for the similar compounds, similar proteins, and whatever the homologs we have, and see that, understand the protein target, and see that how it is made out of, and how you can target it effectively. If you understand it, if you can target, if you can take the right one, right orientation, and include all the points we have discussed so far, we can able to achieve the selectivity to the greater extent. Okay, yeah, that's it from me for today. And yeah, so if you have any questions, you can reach out.
So it's really nice talk from Dr. Sivakumar. So if the online participants if they have any questions, is there anyone online in the YouTube link? So then you can directly ask him, or for the offline participants, maybe you can pass on the questions to me. I'll forward to him for your answer and all.
Yeah,
so he can answer later on also, right? You can maybe by email. I'll be sending some this one. So
yes, I'm sharing the email ID
right?
here. Yeah, the email ID is here. So if you have any notes, you can just reach me. Just training at bicognition.com. You can just write, or you can just visit our website. Just we are very active on LinkedIn, so you guys can check our LinkedIn page. Also, you can reach me over LinkedIn. Also, we are very active on LinkedIn. You can also reach. In case if you are interested to know more on artificial intelligence and drug discovery or CAD-based drug discovery, if you want to learn further or if you want to work with us, so you can also feel free to write to us. We can collaborate, we can work together, or you can learn. Anything is fine. Like we will discuss and then we can take it further according to your need.
Yes. Anyway, again, thanks a lot. Please join me in thanking him again for his 2 days very informative and nice lectures. Hope we'll be organizing some more lectures in future as well. So thanks a lot for joining us. Thank you.
Yeah, thank you, sir. Thank you, participants. Yeah.
So this is bye. So we'll be closing the sessions here, and then the next session will be after the lunch around 2:30. Thank you.